And my name is Shur-Jen Wang, and I'm going to talk about the gene analysis of COVID-19 disease portal. Oh yeah. So Rare Genome Database was established about 20 years ago, and it was a database for model organism rats. But nowadays, up to now, we have expanded to 10 species. And those are the 10 species listed on the top. And today I'm going to focus on disease portal, which is the major piece that links the 10 species together. The disease portal are established sequentially by targeted curation of a specific disease area. Up to now, there are 15 disease portals. I'm going to focus on the COVID-19 disease portal, which is very in time for the COVID pandemic. In COVID-19 disease portal, the manually curated annotation and imported data sets are organized by 9 ontologies. And I'm a curator, so I'm very enthusiastic about ontologies. And each selected ontology can be viewed by the browser in the middle. And here you can select which disease that you are interested in. And in this case, COVID-19 was selected. And down at the bottom, you can send the selected disease genes for enrichment analysis. Again, we have disease ontology, pathway ontology, phenotype ontology for enrichment analysis. And if you want to look at annotation, you go to the orange icon. That icon will bring in the annotation page. On the annotation page, there are options that you can choose human genes or mouse genes or rat genes. And on the top, you have the option to download all the annotations, or you can send the disease genes to analysis tools at the Rare Genome Database. And in this slide, COVID-19 genes were analyzed by their associated evidence code. Evidence code describes what type of evidence supports gene disease association. And the evidence code are listed down at the bottom. These associations are inferred either by genetic association, like IAGP, or direct association, mutant type association, or expression pattern change association. So among these COVID-19 disease annotations, most of the evidence codes are based on expression pattern change, and they are IEP or HEP. And because COVID-19 disease is quite new, just slightly more than 20 years, so most of the genes only supported by one type of evidence code. COVID-19 disease genes not only just associate with COVID-19, it also associates with other disease. So in panel A, we break down this associated disease into high-level disease terms. And based on the number of association genes, you can look at all the breakdown of the disease category. On panel B is the breakdown of developmental disease, and on C is the breakdown of disease of anatomical entity. And we focus on panel C. We break down the top 6 organ systems that have disease associated with COVID-19 disease genes. And on this table, you can see that there are more than 700 genes also associated with neural system disease, and GI disease are followed. And in this table, I would like to point out that the association of autoimmune disease genes in these 4 systems. And this is a confirmation of what observed in clinics. There are a lot of immunodysregulation among COVID patients. So we not only just look at the numbers of genes associated with certain disease, we also take the gene list and do enrichment analysis. And 2 tools are used. One is MOEC, housed at RGD, and the other one is SET Analyzer at Comparative Toxicogenomic Database. And to our surprise, these 2 tools identify liver disease as the highest enrichment disease among these gene sets. And also it confirms that immune disease or inflammatory disease are highly involved in COVID-19 disease genes. We also analyze GO enrichment analysis, and those terms highlight yellow are the top 40 enrichment terms. And you can see that in molecular function aspect, they are highly concentrated on 2 branches. And these 2 branches are molecular function regulators and binding terms. These binding terms include antigen binding. And we also look at the biological process aspect. And you can see that the enrichment biological process terms are also highly focused on some branches. And these are response to stimulus and immune system process terms. So these are the confirmation that COVID-19 disease genes are highly involved in immune function. So in conclusion, we have a timely release of COVID-19 disease portal. We curated publications and then integrate also imported data from other data resources to make this data available to either experimental biologists or computational scientists so that they can use this data for their study. And we perform disease enrichment analysis. This reveals that COVID-19 associated human genes also involved with other disease like liver disease and immune inflammatory disease in several organ systems. And we finally confirm the molecular function by GO enrichment analysis and confirm that the immune system genes are highly involved in COVID-19 disease pathogenesis. In the last slide, I would like to thank our funding agent and also the user that use our database. And please visit our website and give us a thumb up or send us a message. We will respond to you as soon as we can.